This recording is going to cover the organization and basic physiology of the parasympathetic nervous system, which is one of the divisions of the autonomic nervous system. Now, like the sympathetic nervous system, the parasympathetic nervous system is going to target visceral effectors, which include smooth muscles, cardiac muscle, and glands. The parasympathetic division is also referred to as the craniosacral division because of the cell bodies of the preganglionic neurons arise from nuclei in the brainstem, which you see right here. So arise from nuclei in the brainstem. And these are nuclei associated with some of your spinal nerves. And the spinal nerves that carry parasympathetic fibers include spinal nerve 3, here, spinal nerve 7, 9, and 10. So 3 is your oculomotor, 7 is facial, 9 is glossopharyngeal, and 10 is the vagus. But that's your cranial portion of that craniosacral division. The sacral is because of the location of preganglionic neurons arise from the sacral segments of the spinal cord, and specifically S2 through S4. So S2 to S4 segments of the spinal cord. So that's why they call it craniosacral. I do have to remind you, though, that visceral motor nuclei associated with the lateral gray horns. The lateral gray horns are only located in the thoracic and the first 2 segments of the lumbar spinal cord. So there are no lateral gray horns in the sacral spinal cord. The cell bodies, or say the visceral motor nuclei for the parasympathetic division, are along the sides of that spinal cord, the gray matter, but there are no true lateral gray horns. So I do want to mention that. Now, there is, again, a 2-neuron system. You have your preganglionic and your postganglionic, and you're seeing here some locations. You can label these later after I go through it. Is the cell bodies of postganglionic neurons are then are located in these various ganglion, and then the postganglionic fibers will then go to their target structure, and the target structures in smooth muscle, cardiac muscle, or glands, and the effects could be excitatory or inhibitory depending on the specific receptor located there. Now, the characteristics of these preganglionic neurons for the parasympathetic division is the axons of these neurons are fairly long because the ganglion are closer to their effectors. The axons are lightly myelinated, though, just like we had seen with the sympathetic division. The ganglion, we're going to look at different ganglion very shortly, but the postganglionic neurons, the axons of those are fairly short because those ganglion are fairly close to the particular targets. And those axons are not myelinated, just like you saw with the sympathetic. So the main difference when we talk about just general characteristics of postganglionic neurons between parasympathetic and sympathetic is length. They both are not myelinated, but the one is in the parasympathetic nervous system. Those are short. In the sympathetic, they're actually fairly long. Now, the general organization of the parasympathetic system is a little bit easier than the sympathetic, but is in regards to the various ganglion involved. Now, it's again to remind you, cell bodies of preganglionic neurons, they're going to be located in nuclei in your brainstem, or nuclei in spinal cords S2 to S4. Now, the cranial nerves that will be carrying these axons of preganglionic neurons include cranial nerves number 3 here. This one would be 7. This one would be 9. and this one would be 10. And we're going to go through the names of different ganglion and their target structures. So what I'm going to do is I'm going to tell you which names of each one of these ganglion, which what I've labeled as A, B, C, D, E, and F, and the target organs that correspond to these ganglion. So you'll notice the ocular motor nerve is associated with a ganglion that we refer to as the ciliary ganglion. You've seen that term ciliary before when we talked about the eye, because one of the target organs or structures for the ciliary ganglion is the ciliary muscles. So target would be ciliary muscles. So we have ciliary muscles. Remember, they alter the shape of the lens, but also it targets the pupillary radial muscle, which affects the diameter of the pupil. So both of these are smooth muscles. So they're intrinsic eye muscles. Option B, the ganglion is referred to as the pterygopalatine ganglion. That's again associated with your facial nerve. And the pterygopalatine ganglion, that will have cell bodies of postganglionic neurons that will be targeting nasal glands and lacrimal glands, as well as couple salivary glands. But though actually not couple, so I'll just ignore what I just said. So it targets the pterygopalatine ganglion targets the nasal and lacrimal glands. Facial nerve is also associated with the submandibular ganglion, and this will target your submandibular salivary glands, named because literally they're located under the mandible, and your sublingual, because it's located under the tongue. So these are salivary glands, and I'm going to write salivary. The cranial nerve 9, which is your glossopharyngeal nerve, is associated with the otic ganglion. Remember, otic is a term that referred to like your ear. And the reason why it's called otic ganglion is because the gland that's innervated by the fibers coming from the otic ganglion are located near your ear. And that structure is referred to as your parotid salivary glands. So you'll notice we have a couple cranial nerves that will be innervating salivary glands. And just start thinking about it. Parasympathetic nervous system is a rest and digest division. And when you're thinking of digestion, you're going to definitely think of salivation. That's the way to remember it. Now, the vagus nerve is the only cranial nerve that won't be innervating structures up in the head. So oculomotor and the facial nerve were innervating structures in the head, but the vagus, we're going to be going down and the preganglionic neurons will synapse with cell bodies of postganglionic neurons and collectively referred to as intramural ganglion. We're not going to give you any specific names for it. So we're just going to, you see just E as intramural ganglion. But the targets, there's a multitude of targets. So the vagus innervates visceral targets in your thoracic cavity, and abdominal cavity. And you'll see later how extensive that is. So things like your heart, your lungs, your stomach, your small intestine. So there's a lot of different targets for the vagus. Now the last one you see with the F, these are intramural ganglion, but these are associated with the sacral division of the parasympathetic nervous system. Their targets, now the targets from the sacral spinal cord were going to be the inferior abdominal cavity, say for example the lower part of your large intestine would be an example, and structures in your pelvic cavity. But we'll go over specifically targets. But you can see some of them based on the picture that you have earlier in your notes. So I want to do is a little bit of the basic physiology, talking again about the pre- and postganglionic neurons and neurotransmitters and receptors involved. So with the preganglionic neuron, which you see here, those are cholinergic, so that's why you see the C, but also postganglionic fibers, you see here, are also cholinergic. So that means they release acetylcholine. So all this here, that's acetylcholine. Okay, so they release acetylcholine. Now the receptors located on the cell bodies of the postganglionic neurons, you'll notice has the N on it, and those are nicotinic receptors. Okay, so nicotinic receptors are ligand-gated sodium channels. But when we look over here on the targets of, ultimate targets of the parasympathetic nervous system, is here the acetylcholine binds to what referred to as muscarinic receptors. And the response of the target can be excitatory or inhibitory, again, based on the specific subtype of muscarinic receptor. But I want to go back again, just kind of want to mention to you nicotinic versus muscarinic receptors, because both of them bind to acetylcholine. So nicotinic receptors are ligand-gated sodium channels. So here acetylcholine has bound to this nicotinic receptor. What is allowed is sodium to enter the cell, which leads to depolarization, which causes excitation. In this case, we're going to generate an action potential, and that action potential is then going to travel again along this here, the postganglionic neuron, and that will cause release of acetylcholine by the postganglionic fiber. Now, nicotinic receptors, you'll notice, is located at the synapses between the pre- and postganglionic neurons of both parasympathetic and the sympathetic nervous system. But it also, nicotinic receptors we'll talk about when we talk about skeletal muscle physiology. It is the receptor at the neuromuscular junction of skeletal muscle. And it also, the nicotinic receptor is on the hormone producing cells of the adrenal medulla. Now, the duration of the effects by the acetylcholine that binds the nicotinic receptors is relatively brief because it's broken down by acetylcholinesterase. Now, if we have a muscarinic receptor, which I mentioned, this is going to be on your targets for the parasympathetic. Oh, one more thing I do want to mention. The nicotinic receptor is on a sympathetic target, sweat glands. So that's one exception, or actually, that's not actually, delete that. Nicotinic receptors are not on sweat glands. The sweat glands have muscarinic receptors, which is a sympathetic target. Now, so muscarinic receptors are located on all our parasympathetic targets, as well as those sweat glands that we talked about when we did sympathetic nervous system. Now, the muscarinic receptor, we have subtypes of it, but I'm not going to go over 1s, 2s, 3s as I did with the sympathetic, with the adrenergic receptors. But depending on the subtype of muscarinic receptor, the effects could be excitatory or inhibitory. So both examples here, acetylcholine bound to its receptor. Muscarinic receptors are G protein coupled receptors. G protein coupled receptors, again, are examples of metabotropic receptors. So they will have effects on channels, ion channels, indirectly using these second messenger systems. So the example on the left, the acetylcholine binding to its muscarinic receptor, this one's inhibitory. And what has happened is it ultimately causes the opening of potassium channels. So you have a lot of potassium inside the cell. So if you open a potassium channel, potassium is going to want to leave the cell. So potassium leaves the cell, which leads to hyperpolarization, which is inhibitory. On the right, the acetylcholine bound to this specific muscarinic receptor, it could have had a number of different things. It could have closed a potassium channel or opened up either a sodium or calcium channel. So these sodium and calcium will enter. In this case, potassium won't leave. What results from either of these is depolarization, which is excitatory. So we'll give you a couple examples. So a target that would have an inhibitory effect would be, say, cardiac muscle. If the acetylcholine bound to the muscarinic receptor on cardiac muscle, it slows down the heart rate. An example of an excitatory effect would be, say, for example, smooth muscle in one of the muscles in the eye, say the pupillary radial muscle. That caused that muscle to contract, and so that would be excitatory. So if we're talking about smooth muscle, inhibitory effect of smooth muscle, it would cause that muscle to relax. If it had excitatory effect, it would cause that smooth muscle to contract. In the case with, actually I should say inhibitory, I shouldn't say cardiac muscle, because heart rate has nothing to do with the muscle itself per se, is this would be like cells of the cardiac conduction system. Specifically SA node, that caused heart rate to go down. But it also can cause the muscle of the atria, which are the upper chambers of the heart, to relax. So it actually would decrease contractility. So we can have inhibitory or excitatory effects based on the specific type of receptor located on the target. And we will talk about specific effects at a later time.